Welcome to today's lecture. Today we are going to talk about digestion of protein. So, protein digestion starts mainly from stomach. In stomach, chief cells produce pepsinogen enzyme, which is a pro-enzyme, that means it is inactive, which is then activated by HCl, which is produced by parietal cell of stomach. In presence of HCl, there is conformational change in pepsinogen, due to which it leads to self-cleavage and its activation. So, after its activation, pepsin digests protein into polypeptides and few amino acids. When the content moves to duodenum, pancreatic secretion are released into it. Proteolytic enzymes present in pancreatic secretion are trypsinogen, chymotrypsinogen, procarboxypeptidase, and proelastase. First of all, these all are in inactive form. They need to be activated to act on protein. Enterokinase is secreted from enterocyte and it acts on trypsinogen to produce activated trypsin. Then this trypsin is responsible for its self-activation. Then this trypsin activates chymotrypsinogen, procarboxypeptidase, and proelastase and forms chymotrypsin, procarboxypeptidase, and elastase. Due to which finally all these enzymes acts on polypeptide produced by pepsin into oligopeptides and few amino acids. Now the content moves to jejunum. The luminal membrane of enterocyte have aminopeptidase. The aminopeptidase act on polypeptide produced over here to form 3 amino acids, dipeptide and tripeptide. In this way, digestion of protein occur in human. So talking about final digestion and absorption, it happens inside the enterocyte. So let's look how this dipeptide and tripeptide get finally converted to amino acid. Okay, so this one is an enterocyte from jejunum, and we have enzyme over here, aminopeptidase, that digests oligopeptide to tripeptide and amino acid. Now these are absorbed into enterocyte. So amino acid gets absorbed with the help of a sodium dependent pump. Okay, which is called solute carrier, SLC, and amino acid moves along with sodium. Okay, this is also an example of secondary active transport. Now what happens to di and tripeptide? So di and tripeptide moves with proton. So this is example of proton coupled peptide transporter, EPT1. Okay, in this way amino acids di and tripeptide are absorbed into an enterocyte. So here we have enzyme peptidase that will digest finally di and tripeptide into amino acid. In this way all of the protein are digested into amino acid, which is the simplest form. And from here it enters blood circulation via facilitated diffusion. So we saw how protein we take in our diet gets digested at different level: stomach, duodenum and jejunum, and finally into enterocyte to their simplest form, that is amino acids. So this was all about today's lecture. Thank you for watching it.